Hay personas que nacen sabiendo que jamás podrán estudiar dentro de un edificio, porque lo que les apasiona está afuera, en las plantas o el suelo. Sus referentes también están afuera, contribuyendo al desarrollo de una agricultura sustentable y preparada para encarar los nuevos desafíos que enfrenta el planeta. Desde 1876, la Universidad de Chile mantiene este compromiso, el de avanzar el mundo en distintas áreas, generando avances notables en producción e investigación. Y no es porque la Universidad de Chile sea la universidad tradicional del país, es que sus más de 100 docentes también son como tú. Cuando ven una manzana, un pez o un suelo, ellos piensan más allá, porque sabemos que ya no basta con producir más alimentos. Estos además deben ser saludables, sabrosos y su producción debe ser de manera limpia, preservando el ambiente y los recursos para las futuras generaciones. Un futuro agrónomo o agrónoma no debe tomar la decisión de estudiar la carrera sin antes conocer Antumapu. Los invitamos a que vengan al campus, que interactúen con otros estudiantes y que recorran todos los espacios del campus habilitados para la formación de un profesional que nació para mejorar la calidad y los procesos de producción, para transformar de forma consciente y sustentable los productos agrícolas y alimentarios. Estudia agronomía en la Universidad de Chile, acreditada. Hay personas que nacen sabiendo que jamás podrán estudiar dentro de un edificio, porque lo que les apasiona está afuera, en las plantas o el suelo. Sus referentes también están afuera, contribuyendo al desarrollo de una agricultura sustentable y preparada para encarar los nuevos desafíos que enfrenta el planeta. Desde 1876, la Universidad de Chile mantiene este compromiso, el de avanzar el mundo en distintas áreas, generando avances notables en producción e investigación. Y no es porque la Universidad de Chile sea la universidad tradicional del país, es que sus más de 100 docentes también son como tú. Cuando ven una manzana, un pez o un suelo, ellos piensan más allá, porque sabemos que ya no basta con producir más alimentos. Estos además deben ser saludables, sabrosos y su producción debe ser de manera limpia, preservando el ambiente y los recursos para las futuras generaciones. Un futuro agrónomo o agrónoma no debe tomar la decisión de estudiar la carrera sin antes conocer Antumapu. Los invitamos a que vengan al campus, que interactúen con otros estudiantes y que recorran todos los espacios del campus habilitados para la formación de un profesional que nació para mejorar la calidad y los procesos de producción, para transformar de forma consciente y sustentable los productos agrícolas y alimentarios. Estudia agronomía en la Universidad de Chile, acreditada. Hay personas que nacen sabiendo que jamás podrán estudiar dentro de un edificio, porque lo que les apasiona está afuera, en las plantas o el suelo. Sus referentes también están afuera, contribuyendo al desarrollo de una agricultura sustentable y preparada para encarar los nuevos desafíos que enfrenta el planeta. Desde 1876, la Universidad de Chile mantiene este compromiso, el de avanzar el mundo en distintas áreas, generando avances notables en producción e investigación. Y no es porque la Universidad de Chile sea la universidad tradicional del país, es que sus más de 100 docentes también son como tú. Cuando ven una manzana, un pez o un suelo, ellos piensan más allá, porque sabemos que ya no basta con producir más alimentos. Estos además deben ser saludables, sabrosos y su producción debe ser de manera limpia, preservando el ambiente y los recursos para las futuras generaciones. Un futuro agrónomo o agrónoma no debe tomar la decisión de estudiar la carrera sin antes conocer Antumapu. Los invitamos a que vengan al campus, que interactúen con otros estudiantes y que recorran todos los espacios del campus habilitados para la formación de un profesional que nació para mejorar la calidad y los procesos de producción, para transformar de forma consciente y sustentable los productos agrícolas y alimentarios. Estudia agronomía en la Universidad de Chile, acreditada. Hay personas que
Thanks.
You're more than welcome to, come join our program for a few months in the summertime. But a lot of the things that we do are not done in a vacuum. They're done because of students that work with us. So okay, that works. Okay, wonderful. So these are some of the students that have come through the program. Maybe you recognize Stefano Uzaki. He was with us in 2003, 2004. So yeah, I assume that all of you know what rootstocks are, correct? Yes. Okay, so I don't have to go through this. Okay, I can skip this. Okay, and I can also skip this because you know what grafting is, right? And you know what nursery is, right? Okay, I can go forward with this. But what people sometimes don't realize is how central the rootstocks are to the whole orchard system. It used to be, and hopefully not anymore, but it used to be that people didn't think much about rootstocks. They said, oh, what variety do you have? You go, oh, I have this super maxi gala, astronomic. And you go, okay, what about the rootstock? And I said, oh, I don't care. Just whatever, as long as I have the variety. And where actually it plays a very, very central role with all of these interactions with the soil, the science, and the type of culture that it has. And that's it's a difficult item to overcome, this notion that rootstocks are just for whatever. They don't matter for food production. This one of the reasons why was initially the dwarfing rootstocks were implemented in Europe, and I will talk a little bit more about that. But this is true dwarfing. Okay, these two trees were planted at the same time. Okay, same sign variety. This is Matsu. But one is producing a lot of wood, the other one is producing fruit. And the difference is the root system, the rootstock. This is where we used to be 100 years ago. In fact, this picture I think is 1901, and this is where now we are in many of the orchard in the U.S. Some or one form of this, or at least a more of a 2D system. But the question that I have is, how old is M9? You know M9, right? How old is M9? How long has it been around? Huh?
100 years.
Okay. Perhaps that's 100 years name under M9. How old? How old do you think?
110.
Okay, so this is 1866. And we're already talking about paradise stocks in England, okay? Or Duchenne stocks. So the paradise is associated with M9 or Duchenne. Okay, so that's 1866. It was around back then. 1767. We can retrench. Okay. The premiere for graph, and then so on and so forth, and then that for Duchenne. And so you find Paradis and rootstocks in 1767. I can go back a little bit earlier, probably around the 1600s. But M9 technologies has been around since basically the 1600s. And this man right here did basically all he did is took all the rootstocks from around the German and French and Belgian, Netherlands and England, and essentially characterized them. He did a great job. So we owe it a lot to having, this revolution of rootstocks like M9 to run happen. But our story, the story of the Geneva rootstock, starts pretty much when I was born, 1968. Okay. And Dr. Cummins and Alwinkel, we had a problem with M9. It wasn't resistant to fire blight. In fact, it died of fire blight, and we lost many trees from fire blight. And so they decided to focus on disease resistance, fire blight, and Phytophthora. And then also, obviously, the Cummins program was dwarfing and precocity. And that program went on until I joined. We joined up in this time. That's when Cornell made the decision to stop the program because it was costing too much. They didn't want to replace the people. But the industry said, they went up to the government and said, we need to continue the program. So that's when the USDA joined the system. And at that time, so right around this time, we had probably a production of about 100,000 Geneva rootstocks per year. Right now is about 15 million trees a year that we produce on Geneva rootstocks. And I'll show you why and some of the things that are unique to the Geneva rootstocks. And and these are all the new releases. These are the newest releases. So there is a method to the madness that we have, or that I have. And it is madness because it is meant to work on root systems. And this is basically describes all the protocols and things that we try to do before we release a rootstock. But look at this. These are stages, and it takes about 27 to 30 years to have a good idea about what a rootstock does. That's a long-term breeding program. And that's probably why the USDA joined up. We can do some things with molecular markers. Okay. We can essentially at this point generate a lot more data and a lot more replications. And we do it. Yeah. But we cannot save very much time. Basically, this evaluation time gets moved up over here. The evaluation time in the field in multiple field trials is perhaps the most important asset that we have in the breeding program. And I will show you why. Okay, so yeah, we play around with a lot of genomic stuff, genomic resources. And so we have the largest collection of gene expression markers and RNA-seq data for apple rootstocks. We utilize, the SNP chips, and we have made the genomes. We haven't released our genomes yet. Probably in the next year we'll do that. But those are all, there are tools to the means to an end to make sure that we have the best rootstocks out there. What we like to think about is what can we do for apple growers and to resolve some of their problems, and those problems are associated with money that they get to keep, or they get to save, or they get to make based on those traits. And so obviously, if you have an increase in productivity, that means a lot of money. Woolly apple-leaflet resistance, some money. Fire blight resistance. If you're in a fire blight area, it means a lot of money for you that are being saved because you don't want to lose trees. And and I've seen, and now in my career, 3 major fire blight events that probably cost between 200 and 500 million dollars. Okay, so we're moving away of that. And then, unique uptake. It used to be that in 2001, this was the landscape of the Rootstocks that were being sold. Most of them were M9 clones, then B9s, a little slice of Geneva. And now you have more of a diversity. And having, as you know, having a diversity means that you're able to do many, many things, and not just one thing with one rootstock. And we'll see that. So one of the things that we're able to do is, and this is just an example of just Fuji, Gala, and Honeycrisp. We're able to take some of the characteristics that are in the rootstocks, like calcium to potassium ratio and things like that, and match them to the needs of the scion. So the idea is to create this designer or this matching, better matching of the root system with what the scion needs. So if the root system is weak, we have a more bigger scion. If the root system is if the scion is weak, we have the anyway. You get the idea. And having a more diversity in rootstocks, you're able to build canopies that are very, very different according to different localities. This is our friend. He passed away just a few months ago, Nicola Della Becta. But this was G969. Beautiful trees in Italy with three main branches. So a nice wall of apples, as you see, and it's extremely productive. Okay. So what's going on in the graft union? This is a CAT scan that we did at Cornell because we wanted to understand a little bit more about how the graft union forms and what the shape is of the graft union. So this is a graft union. There's a lot going on. This is obviously the scion. This is the rootstock. And we ask, what's going on? What's happening in this relationship when you're joining two different genotypes? What type of communication happens, and how can we leverage that union in order to create more productive trees and trees that are more resilient? And what can we achieve by separating the thinking about the genetics of the roots separately from the genetics of the scion? So you end up having efficiency because all of a sudden I don't have to worry about how the rootstock fruit tastes or whether it can be stored in the shops or whatever. And so there are a lot of efficiencies that are created by separating the two, the one that grows in the ground and the one that grows up into two. And you have a lot more probability in making good selections faster. So I talked a little bit about this a little earlier. These are examples of rootstocks or the root-induced traits on the scion. Dwarfing and early bearing. We have branch angle modification, increased flowering and bud break in low chill environments, and hormone, mineral, nutrients, and increased whole tree tolerance to diseases. And I'm gonna mention some of those. The first one, of course, is dwarfing. This is dwarfing coming from M9. Yeah, we have to use the microphone.
Oh, it's just for the people in the.
Oh, it's for the people. Okay, I'm sorry. So, what we discovered is that the dwarfing from M9 and also M8, it's a 2-gene model, which is dwarfing 1 and then dwarfing 2. DW1 is on chromosome 5. W2 is on chromosome 11. And based on the combination of the alleles, this is what the alleles are: AA, AB, BB, and then and so on and so forth, you can create different levels of dwarfing, but also different levels of early bearing, or the amount of buds that go from vegetative to flowering or fruit production. And this, I don't know how many of you know what about QTLs are. QTLs, yeah? No? Okay. Simple. These are the 17 chromosomes of apple, and these are the markers that are associated with changes in dwarfing. Okay, so you compare the means, and you get this nice graph. And this is where dwarfing 1 is on chromosome 5, and this is where dwarfing 2 is. Very simple model that we came up with. This is early 2000s. 2 genes, okay, or 2 loci. I don't want to call them genes, but 2 loci, two things. How much are they worth to the industry in the US? And you can estimate how much they're worth. This is, so I talked with Terrence, I talked with Stefano, they said, this is not enough, 61%. It should be 75 or 85%. But they say that 80, well, okay, let's say 80% of the increase that we saw in productivity from 1982, and these are data per hectares, tons per hectare, is due to 2 genes. Okay. At least $2 billion. Worldwide, much more. Probably 10 to 15, maybe $20 billion that are the 2 genes are worth. And we have an idea of what they are, but we're working on that. But it's amazing how the change from 40% of rootstocks that were dwarfing to 93% nowadays has made such a huge difference to the industry. People used to say, "Oh yes, we need to grow big trees and big things," but they didn't realize that dwarfing and early bearing are amazing at increasing the productivity of apples. So our collaboration with Terrence Robinson and many other research scientists, this is what it's all about, field research. And so we have our rootstocks tested, and this is M26 and M9. This is basically the cumulative production, and we do this with many newer rootstocks. But you can see where we're at with some of the most productive rootstocks. We just released this one for Snapdragon, and specifically for Snapdragon, because it produces larger fruits and it produces more fruit compared to other rootstocks. So rootstocks affect yield, but they don't only affect yield. This is the biennial bearing of a rootstock. So sometimes of science, sometimes things like Fuji, sometimes Honeycrisp, they can produce a lot of flowers one year, and then all of a sudden, next year they don't produce any flowers. And it turns out that some rootstocks are able to bring that fluctuation down. It's an interesting mechanisms that we can talk about it later on if you have questions. And then you have a rootstock effect on the Purgon lanigero, where you have resistance, and if you deploy the resistance to the Purgon lanigero, essentially you eliminate the source of the Purgon coming back in the springtime. And that has worked. We have many orchards now that don't require spraying for the Purgon because of the presence of resistant rootstocks. And so every year, this is a photo taken maybe a few months ago in our greenhouse. you can see we're testing the foregone and whether some of our rootstocks are resistant or like that, and we put them under very, very high pressure. We also have resistance to fire blight. This is what happens. This is an M9, but fire blight can be devastating. So this is an example how devastating fire blight can be. This is a test orchard that we planted, and we planted our rootstocks like randomly inside the orchard, one tree at a time, not groups or whatever. So this is a completely randomized experiment inside a Gala M26 orchard. This is coming back after 2 years, and about 70% of the trees were gone, and all of our rootstocks were, all the trees on our rootstocks were alive. So, you know, imagine being the grower that, well, they had to take this orchard out. They came out and then they took all of our trees and put them all in a row, and they transplanted those, and they were happy. So fire blight. Another thing is I talked about chilling requirement. And we know what's happening with global warming or with climate change. Let's call it climate change. Some things, some places are not receiving the number of chilling hours that are needed. As it turns out, one of our rootstocks, and this is work done in Brazil, you can see the difference in bud break between G213 and M9. And this has been repeated year over year over year. That difference means more flowers open at the right time and also more yield. And it's done by the rootstocks. We don't know exactly what mechanism. We have an idea, but we, you know, every mechanism has an hypothesis. But, you know, how does the rootstock do that? We thought that chilling was set by, you know, the genetics of the scion variety, and there was no influence from the root system. There is influence. And I was saying that one of the benefits that we had, one of the great things that I enjoy as a breeder of rootstocks is the collaboration with the NC140 people. You know, you've met some of them, many of them, and some of them have been, I think, here working with cherries, because we don't only do apples, we do, you know, cherry, peach trials, pear trials, all kinds of things, and this data is available to all of you if you need it. You just have to go online and download the papers and things and see what rootstocks perform well. But it might not work for you because you're at very different environments. This is why we have this testing of rootstocks throughout all the United States, is so that we can identify which rootstocks are best in certain environments, but also which ones are more stable in all the environments. So releasing or preparing an apple rootstock to release requires long experiments, decades. So when we look at all of these titles and things that graphs that we produce, sometimes they represents 10, 12, 15. One of the latest papers was 20 years at, you know, harvesting all the apples and counting and measuring the fruit size and all of that. And so the key word becomes long-term performance, you know, and so on and so forth. Because why? We're dealing with rootstocks. Once you put it in the ground, and you need to expect the lifetime of an orchard in order to properly evaluate. And what we found actually is that sometimes at the end of the or life of the orchard, things tend to change. Some rootstocks that we thought were awesome went up. Some went down. And so beyond productivity in all these studies, we started noticing that rootstocks were changing the quality and the properties of the apples that we were eating. This was largely ignored in a long time because you're only using M9 or using one rootstock. You don't see that unless you use a diversity. And so we think about the changes in tree architecture and all the other things and how they affect apples. This is what we found. Okay, so each one of these dots, okay, let's see if I can explain. Each one of those dots is the mean. So it's a genotypic mean of the rootstock. Okay, so these are 160 rootstock genotypes. Okay, so for each genotype, rootstock 1 to rootstock 169 or whatever, that's the mean of 10 to 7 tree annual data for bricks. So we measure the average, and so that average for that rootstock. So it's a very, very solid number. You can see that bricks firmness have a very, very strong relationship, and there's not much a relationship with that tree size. So there's something else that's going on beyond tree size that's affecting bricks and firmness. And then we look at this is a different experiment, and we're looking at bricks, and you can see that some rootstocks were influencing over, this is 2015, 2016, so 3 years, over 3 years, we're influencing the bricks or the sugars or the maturity, you can see, of the apples. So yeah, rootstocks affects maturity or sugar. What else do they affect? Well, look at that. This is work by Jaume Lordan, who is in Spain. This is an example of the effect that rootstocks have on endogenous hormones collected from the xylem of the scion. Okay, so this is, I think this was all Honeycrisp scion. So we sucked up scion and sent it to a special lab to measure the hormones. We measured all, the whole profile from cytokinins and things like that. And this is what we saw. Okay. And this is just ABA as an example, but huge differences in ABA. What does ABA do in a whole tree? Some things, maybe it shuts down the stomates, maybe it changes the metabolic but it's pretty powerful. How powerful is it? So this is an experiment, and it's the first of its type, I think, in the whole literature, showing that the rootstocks were changing the gene expression of grafted scion. So it changed. So the root systems are able to change the intrinsic way that the scion are expressing their genes, the amounts. And so this is an experiment that we did comparing dwarfing rootstocks versus non-dwarfing rootstocks. A long time ago. This is 2010. So experiments were happening in 2007, 8, 9. And so with those changes, you also see changes in the number of feathers that the rootstocks are doing and how big the trees are in the nursery. Also, and you see that like that. And this is a funny one. So this is a field trial that was in South Carolina, and I was noticing that only one, so it was Fuji, grafted on 10, 15 different rootstocks, and only G16 was being pecked on. Only one rootstock had the holes. You know what the holes come from? We have woodpeckers here. So only one rootstock was giving some type of, I don't know, juices or whatever. And then I saw the same thing in Maine and on an island in Maine that, where trees were being pecked by woodpeckers. But the rootstock was, I don't know, maybe making the thing more sugary, or I have no idea, but it was only on one. And so you can see how rootstocks also can affect the color and the size of apples. And sometimes in a very dramatic way. So this is an example of statistics and things like that. We released this rootstock because it has a very, very real effect on fruit size, even with the same crop load. Mechanisms, we can talk about that. And then we do a lot of research. So we measure things in the fruit, but we also try to understand what roots are doing. And this is some of the work that we do with mini rhizotrons. This is a long time ago when we had to stitch 67, a lot of images in order to find out what the roots were doing. And this is the type of work. He just graduated with his PhD. So this was he was a high school student at this time. And then, after studying the roots for a little bit, he said, wait a second. Roots are, they take up water. They live in the soil, but they also take up nutrients. And we said, how about we measure the nutrients in our populations, in the leaves of grafted science, okay? So we started measuring that, and we saw that in terms of the potassium, the content of potassium in the leaves on grafted science. So I like to measure always on the same scion, because that takes away any differentials that are typical due to the genetics of the crop. We saw that nutrients were being changed by the rootstock. And this is a little example about how some of the nutrients, some of them enter actively through channels. Some so that's potassium has both, has active transport and also passive transport. There is sodium that only has passive. Calcium is a little bit more active. So everything is a bit different. And so we started to think about, what are some of the models or things that are affecting the absorbance and then the transport. So there are two things: the absorbance into the roots and also the transport into the sign, which I call conductance. So absorbance and conductance. And so we have channels, chaperones. You have all of these proteins and things. And then here there are interesting things: root turnover, or the root half-life, or the root expiration index. So how many roots are in the soil, and then the symbiotic or pseudosymbiotic relationship with bacteria, and things like okay. Mycorrhiza. It's right there. Yeah. So association with mycorrhiza and things like that. And those are all real things that affect this. And so I think we were the first group, and I know we were the first group to publish on this, the genetics, how complex the genetics for nutrient absorption and conductance or transport to the scion. And you can see it's pretty complex. Some things overlap. Anyway, and you see some similarity among nutrients. So for example, potassium is very often associated with phosphorus, calcium, sort of in the same group, and so on and so forth. And that sometimes can give some interesting hypotheses. So that was the genetics was done there was an experiment, I think, over 2 years. So we looked at this in experiments over many years. Now this is over 5 years, I think. And you can see, for example, for boron, it's very, very genetically controlled. Some rootstocks are very bad, like M9 and B9, very, very bad at absorbing boron. That's why we need to spray boron. But do we need to spray boron if we have G935? No. It's probably why G935 is more productive. It's part of that whole picture of the productivity. It's the effects of the boron that have on the flower setting, and so on and so forth. Phosphorus, similar story in phosphorus. Calcium is a little bit different. Calcium is finicky because 1 year is one way, 1 year is the other way, and it's affected by climate a lot. Yeah, you go like, yeah, that's true. So, and we produced, I didn't put some of, yeah, anyway, if you looked at the papers that we produced, there's things about the timing of calcium absorption that are interesting. The most important thing for us, for breeding, and we do this using this instrument. It costs about $55,000, but it's worth more than that because we don't have to send material to the laboratory and pay $20 per sample to analyze, and we can get the potassium to calcium ratio. So it's a relative measurement. Doesn't matter how much content it is, but we can get this figure in 35 seconds on an apple. Then we can take that apple, we can store it, and we can look at bitter pits if there is bitter pit, or if we can look at other things. Extremely useful instruments. And the other thing about it is that this is bitter pit, and it's very strongly, this is an R of 0.86, very strongly associated with the appearance of bitter pit, the ratio. And that's been proven. So ratio is potassium to calcium, but we also have genetic effects on the absorption of nitrogen. And so I'm showing here two genetic loci that affect nitrogen. We don't need to know more about that. But the fact is that based on the knowledge that I have about the alleles of those loci, I can combine them, and I can say, well, this is the best, this is the middle point, and so on and so forth. And I can select the level of nitrogen. And does it work? Yes, it does. These are two full sibs. So brothers, or in the rootstocks. And this is a full field. And the changes is G214 is on one side, it's much lighter, low nitrogen. 213 is on this side, high nitrogen. And it works over and over and over in other parts of the thing. Okay, so rootstocks absorb nutrients in a different way. All right. But do they always act the same? And this is a work that we did. Were you here when we were doing the pH work in 2000? This is 2008. So this is, for example, G41, and this is phosphorus. And now we're looking at phosphorus concentrate or content in the leaves of grafted things of trees at grown in different pHs, 4.5, 5, 5, and so on and so forth, up to 8.5. And so you can see that some rootstocks have a nice, interesting curve. Some really don't care. Some others, again, don't care. And 111 sort of is like that. But then we find out that G41, and you'll see in the picture that I'll show you later, G41 really suffers at low pH. Doesn't grow well. But this was the beginning of understanding that there is yet another variable that changes the whole landscape of nutrients. And so we built these rhizotron systems like that to look at how the roots were growing. This is actually a pH experiment. So all of these were randomized with different pH treatments. And we repeated the work. And this is what we were doing. We were looking at how much the roots were growing, how well, and so on and so forth, and how well the trees were growing. And what we found was, of course, the pH was affecting the growth of the trees based on the rootstocks. Let's move on to living things and the interaction of the roots with the living things. And these are pretty much the culprits for replant disease. And what we found was that some of our rootstocks were more resistant than others to replant disease. Okay. Productivity overall. In some cases, there was a 34, sometimes even 50% increase based on if they were fumigated or fumigated. And whereas the genetic resistance provided that resistance overall throughout the life of the orchard. And that was money. It's productivity. And it wasn't just true in North America. This is Brazil. Same thing in South Africa. Same thing in many other locations where we tested. For some reason, some of the same nasty bacteria and whatever are doing the same things to apple orchards worldwide. And so we spent a lot of time looking at the relationships of microbes and why our rootstocks are more resistant and so on and so forth. And so you see even other groups, this is from Germany, looking at our rootstocks, and they found the same things happening, compared to that. And then when we look at just one, Pythium, the resistance to Pythium in our breeding program, you can see that there are multiple. It's a complicated mess in terms of genetics. Oh goodness, this was a great experiment. So people think that roots are all the same. They're not. And this was one of the things that we began looking at what's going on in the root systems in terms of metabolic compounds. And we looked at resistant and susceptible rootstocks and the amount of metabolic compounds. Found benzoic acid was perhaps a target for market development in our breeding program for providing some resistance to I think we were talking about rhizoctonia in here. But actually, and it validated the fact that some of these compounds were associated with good vegetative growth. So we went, and I said to myself, I don't want to dig roots. That is the worst thing. And so I said, how do I get to the root system without having to dig roots and still have some pretty meaningful genetic and physiological ways to do it? And so I built a what's called a system that uses aeroponics. So the roots are growing in the air and they're sprayed with nutrient solutions every 2 minutes. And this is an example of that system. And then we can get nice plants growing like this. So right now the plants that I have in the greenhouse are about this tall. Yeah, about this tall. And so we can do a lot with that system. We can look at mineral nutrients and salinity. We're doing salinity and drought right now. And it's a beautiful system. And people say, oh, well, but it's not the same as in the field. Yeah, but try doing it in the field. So and I think it has a lot of correlation and connections. So we get some nice roots, and we take those, and I can snip these few roots and put them in liquid nitrogen, and I can find nutrients. I can find nutrients, gene expression, and then I can also find hormones and metabolic compounds. Okay, all from the same thing. And then if I look at a spectrum of rootstocks like 15, 20, or 360, like I'm able to do here, then I get more interesting things. And so, okay, these are some of the phenotypes that we see. But inside the root, there is phenotypic diversity for metabolic compounds. Okay, so these are what we're looking at here is these are genotypic means for things like benzoic acid, isoquercetin, caffeic acid. Okay, there is diversity for that, and so from there you can come up with this. And so all of these are different individuals that we tested. All of these are means, okay, for those individuals for these compounds. And obviously you have classes of compounds that are similar, so same pathway and so on and so forth. And if I look at isoquercetin, for example, this is where it's at in the process, and it's related to what is it, rutin and some of the other, and quercetin and so on and so forth, and negatively with some of these other things. So you can start understanding the pathways and the physiology about and the biochemistry about that. But I can go a little bit further, and I can find the QTL for isoquercetin. So it's on chromosome 17. Okay. Well, okay, some chromosome 17. So what? Is this bioactive? Probably. It has some meaning. But having the QTL, then I can do the effect for marker prediction. I can find markers that are associated with that, and then I have RNA-seq for that same root that was harvested at the same time as we measure the metabolic compounds. Guess what? The first gene that come out for in a predictor screening in our thing. It's this guy. Okay, and so it says, what is it? I think I get it in the next. It's this gene right here, and you can find it also in the gene expression QTLs, and it's a UDP glycotransferase superfamily protein. So I can do this based on the RNA-seq experiments that I'm able to leverage. What does this mean? Maybe I know something, maybe I don't. So, but I know that now isoquercetin is affected by this particular gene. And I can make markers for it and select for it. And I can look at the diversity at that specific locus to see what the genome is like, whether there is an interruption or in the gene and the promoter region and so on and so forth. And then I can make markers for that, that basically look like this or look like that. And then I can say, well, if I want plants with high isoquercetin, I'm going to select this group. Going back to the pH thing, we can also do pH work in aeroponics. And you can see here, G41 is expected to grow as well as all of these, but it doesn't at pH 5.5. It's suffering. So this is like the fourth experiments where we confirm that. And we have different ways to measure things. I don't show the artificial intelligence stuff to look at the root pictures and transfer them into numbers. And then we also have had to do some work with viruses. And this was something that happened in 2015, 16, where we started seeing some trees of royal red honeycrisp on G935 doing this. They basically were sitting; they were not growing. And we wanted to know about, why, what was happening. This was a virus issue. This is another example of a virus issue with one of our rootstocks and with stem grooving and stem pitting on Grammy Smith. And you can see some of the escapes right here. The escapes are probably, as you take the buds from the budwood, some buds maybe have less virus and they end up at the very top, and maybe these are the escapes. But it's a problem. It's a big problem, or can be a big problem. So we did experiments with the aeroponic systems, comparing inoculated and non-inoculated rootstocks with this, with the same variety, same sign variety, one that had viruses and one that didn't. And so this was a very strong replicated experiment. This is an example of that. And this is G935. And you can see that the one with the virus, here it is. The one without the virus, that's clean. It's nice. And then some of the other rootstocks are not being affected as much. Another example of what we use these aeroponic tanks is to monitor water use that's influenced by the rootstocks. So I built these dendrometers, and they're hooked up to some very sensitive data loggers. And this right here is the light that is being measured. I think the sensor is up here. And this is the change in expansion. It can detect about 10 micrometers, 10 micrometers. So expansion and contraction of the tree base, and you can see that's very well associated with the light. So at night, you have expansion, and then during the day you have contraction. You can see a little bit better. I think I have it right here. Yeah. So this is inside, what the roots look inside. And again, this is the light available. This is during a cloudy day, and you can see how the plants are reacting. And we also have a video of that, and you can also see the plants, the trees or the leaves going like that, opening up during sunlight, like that, shutting down. And it's a system that's very, very responsive. These are 15-minute intervals, but like if you have a cloud stopping the sun, you can see it react very, very quickly. So we're studying the effect of the roots, like the roots that these two root system genotypes are not behaving the same. And then we can do it for many, many days. This is a day thing. And this right here, these are many rootstocks. And this right here is when we stopped the water. And we did an instant drought for 2 hours, 2 or 3 hours. And then we look at how the trees recovered. Do they keep on growing? Did they stop growing? And things like that. So, yeah. So we have lots of plans in the future. And, we keep on working with Mrs. Stefano Musacchi in Washington. So we keep on working with people everywhere in the world. Hopefully, we'll start working again here in Chile, doing some collaboration research in Chile. But this is somewhat some of the impact that the Genevas have had in the U.S. This is a whole nursery full of Geneva rootstocks, exclusively Geneva rootstocks, and this guy was a student of mine a long time ago. But our our rootstocks have also changed the way people need to propagate them, and sometimes they do cuttings, which was unheard of for apples a long time ago. And then trees are also being produced by containers. And this is another stool bed. I think this is a 214 stool bed. A lot of production though and we've received a lot of help from tissue culture and micropropagation of the rootstock. So we produce millions of those, and these are some of the interesting trees that are being planted. They're less expensive. They don't get you to production as fast as a well-feathered tree or a tree that has many branches, but some people don't want to afford that anymore. And if you come to Geneva and it's between May 1st, May 1 and October 31, and it's a good day, I will take you sailing, okay? It's a promise. I mean, if I'm there, I'm not traveling or anything. So, thank you for listening to me. This is a group of wonderful people that work with me in the past. And, this is what we do. Thank you. Good class. Is that how you say questions? Yeah.
Yeah. If you think about that from the rootstock will have influence in the quality of the food, have to go to the science. So what are the genes? You mentioned something that some studies are there, but how the genes from the rootstock will have influence in the quality of the food on another side, or what genes you can look into that?
Okay, so I think we need to look at the product of those genes as metabolic compounds. So we haven't found very much evidence of RNA traveling that far.
But so far it's evidence that RNA can move.
Yes.
From the rootstock.
Yes. Yeah, it can. Yes. Yeah, and that's probably how. But different RNAs have different distances. So, and we haven't gotten there because there could be chaperones that are shuttling that RNA through the cells, and specific RNAs like microRNAs. I can see microRNAs, and we do have evidence of. I didn't show this when we did the EQTL study. There are specific loci that are changing a major amount of genes from based on the allele that we have. So gene expression turn on or off, and I think about those as being activated by microRNAs. Okay, so probably likely, but then hormones, big huge effect from hormones, and then we don't think about the physiological effects of nutrients or the availability of nutrients. So if something has nitrogen, or if you get more nitrogen, you're going to activate more pathways that say, oh, I feel good now. I have nitrogen. I can, do a few more cell divisions or do like that. And so there are changes detected by the cells of the cyan that are due to just physiological things. So a lot of ways that things can happen to the fruit or to the growth of the cyan. And each one, so if you look at just one thing, it's probably going to be multiple avenues that maybe affect the same thing additively or whatever. So, and you need a very big experiment in order to, you know what I'm talking about. So I haven't done that big experiment yet. Maybe in 2 or 3 years, I will decide to do. So the experiment that you would have to do, as I'm envisioning, is selecting like 4 or 5 different groups of rootstocks Still segregating genetically and all of that. And then grafting the same scion, or maybe 1 or 2 or 3 different scions. So maybe 150 rootstocks, 3 scions, 4 trees for each thing. So now we're 1,000 some, 1,000 plus trees. And then collecting the RNA from the scion at different time points of the growth. So, and then collect the RNA from the apical meristem, which we have done. I mean, we've done it at a smaller scale. So apical meristem, leaves. We've done the layer between the xylem and the phloem. Cameo. Yes, we've done cameo. But in doing it, at such a large scale, then you can get an idea of, based on what genes are being up, down regulated. Because the genes can tell you, the RNA-seq can tell you a lot. If there are pathways that are activated by hormones, which ones are activated by ABA. So you look at the ABA metabolism, genes, and so on and so forth. And so genes can tell, the gene expression can tell you a lot. And so you do that experiment, boom, you understand now the communication from the rootstock to the scion, but it's not one way.
Yes, it's possible.
It's both ways. And that other experiment has not happened.
At the beginning, the program was very well oriented to Phytophthora. But are you still looking for the Phytophthora system?
We do Phytophthora and Pythium. Phytophthora and Pythium are the 2 big ones. Rhizoctonia is number 3. And we try to evaluate that as well. It's in specific situations, but Phytophthora and Pythium are, and I didn't show the picture of the dead, inoculations that we just did 5 months ago. But yeah, we look for kill. We basically, out of 10,000 seedling, we can kill about 5,500 seedlings with just Phytophthora. So, and that's how we select, because it's a very complicated system genetically that we see. So I showed resistance to Pythium. The resistance to Phytophthora is on chromosome 5. And it's a major gene, so it's a little bit easier to select. But, if you want to select and load, that's what you have to do. So we kill.
It's better to kill.
It's better to kill. And then if there are any escapes, we try to catch them, hopefully with molecular markers. But the escapes also, they get caught in the evaluation, during the evaluation, because we put material into replant soils so we can be best. And before we send anything out, it's been evaluated in multiple orchards and things, and that essentially you come to know that it's replant tolerant. 20 years ago, I went to Badensville in Switzerland. I've been there. It's their research station. They had a field trial of G41, M9, Bud9, M26, and G41 was much bigger than the M26 trees. And I said, you have major problems with replant disease. I said, no, we don't have replant disease. I said, you do. And that's the telltale, is where M26 is one of the worst for replant disease. And you can see it in the data. So we try to do both, and then also like that. We haven't done much though with nematodes. Some people have done evaluated roostocks from nematodes, and it's you know, on and off, and then there's an issue with terminal rig spot fires in some places, so not very common, so I don't have time to read for that, not yet. Good? Alrighty, clear as mud. Okay. Okay, it's time for me to go to the airport. Thank you for letting me speak to you. Appreciate it. Can I take a picture of all of you?
As we say in Bali,
La foto.
like that. Beautiful. Thank you very much. Oh, gracias a ti.
And if you have more questions and things, you can just email me. I'll be happy to try to point you in the right direction. You should have a root, you should have a root research program here.
That's the deal.
Yeah. No, it's not worth it. It's too expensive. It's too expensive. It's not worth it.
Bueno, le agradezco que hayan venido. Charla, charla. Muchas gracias.
Yeah. Yeah. This one. Till next time, right?
Next time.
Yeah.
See you again.
Thank you.